It's a pleasure to have Nir Barzilai here today to give Barshop seminar. Sure many of you are already familiar with Nir, but if you are not, you can see that he's the director of the Institute of Aging Research at Einstein. He's the chair of aging research. He's the PI of the Shock Center for Aging Research at Einstein. And what he doesn't show is he still does clinical work as well in the diabetes clinic and still is an attending physician. So I think we're very lucky to have him today. He's got a lot on his plate. For those that aren't familiar with Nir, he started off with medical research in the Israeli army as a medic instructor and a medic, and rose to the level of chief medic of the Israeli army as well. And so after that period of time, he went and got his MD at Technion in Haifa, Israel. So he's the second in a row of Israeli researchers we've had for Barshop that went to Technion and worked there. I won't go too much into this, but I just wanted to point out that, I mean, in addition to all the stuff that he does in his lab right now, I just want to point out that is an active clinician, and what he was doing in the past, he actually helped create some community health projects in South Africa and medical aid to Cambodian refugees. So he really is a medical scientist, I think, and really cares about both sides of it. As many of you already know, there's basically sort of three areas that Nir works on in the lab, and the first is the biology of aging, basic biology of aging, how mechanisms of, what are the mechanisms of aging, how does calorie restriction work? The second is energy metabolism and aging. And the third is sort of looking at the longevity genes among the supercentenarians, those that live 100 years plus. And he's very well funded for each of those projects, and I think his talk today is going to sort of touch on a lot of those, it looks like. The last thing I wanted to say is that he's actually been very, very good no matter what. So I think in addition to be very, very good to talk to no matter what, in addition to being a great scientist, he's very easy to talk to, very willing to help. So if you get a chance, yes, but I need help in the future. So with that, I'll let Nir get to his talk to change the title.
Thank you very much. I have to say that when I listen to this introduction, all it does for me is makes me feel old. You know, it's what you counted is time. You know, is only time. Talking about time, I just want to acknowledge it's a special honor. It's an honor anyway, but it's a special honor for me to be here and have Ed Mazzoro sitting here. Talking about people who don't change with time, I think Ed Mazzoro influenced the aging thing, and I don't know what you found that kept you like that. You haven't shared it. It's not caloric restriction, right? So my title is different from what you have, and you know, forget the title. What I really want to do is tell you some very interesting stories. They'll be connected to each other, and I'm certainly planning and welcome interaction. So I'll be flexible. It's possible that you'll ask me a question and I'll say it's coming, but otherwise we can do whatever you want. I had 40 slides, which sounded good for the time allocation. It usually takes me 40 minutes uninterrupted. I added several more slides. And my excuse is that there are people here that are not mainstream in aging. But it's really not the whole truth. It's something that is introduction to aging that that happened today again and again, and I just want to emphasize that. And if you want to stop me after this introduction and provocation, we can talk a little about that. But I think it's important, whether you're in aging, whether you're not in aging, but especially if you're in aging, to kind of talk of the impact of aging and why is it so important and where do we think it's heading, at least in the leadership view. And I want to start by asking you this question, and I'm going to take a vote. And there is a reason for why I'm asking you. I want to ask you if you think that we humans age at different rates. In other words, do you know somebody, let's say, who's 50 who looks more like he's 40? Do you know somebody who's 50 who looks more like he's 60? If you do, raise your hands, and everybody looks around. So, thank you. So, first of all, this is a demonstration of a question that a professor of university should not ask. We ask questions in order to divide the students, not in order to unite them like that. But I think it's really so important, and I'll tell you, it never fails in any audience, lay audience, scientists, it never fails. And I think it's incredible because intuitively, we all understand that there's a discrepancy between our biological age and chronological age. And I think it's very important to understand that on the animal and then on the human level. And this maybe explains to you why I'm doing all the research of centenarians, which I'm not going to talk about. There's maybe one slide that I'll mention recently, but why we picked centenarians, because we think that their aging was probably, well, or for a lot of them, probably their aging has been delayed by a long period of time. So they're the people that are enriched with the biggest differences between chronological and biological age. But because I'm not talking about centenarians, I really want to have you remember your answer in the context of this slide. And this slide is a slide that is from the government, from the NIH, from the NIA, and that's why it's not a good quality slide, okay? And I'll need to improve it, okay? And what you see here is the relationship between death from diseases, and there's lots of diseases here, and age. And the red line, I think I have this. And the red line is cancer, the blue one is cardiovascular, diabetes, and Alzheimer. And I think what's important to note is that as we go from here 30 to 85, the risks of death goes not by 10% or twofold. It's from 1 or 10 to 100 or 1,000, okay? So, you know, everybody knows that cholesterol is a risk factor for heart disease. And it's a threefold risk factor for heart disease. But it's nothing compared to aging, okay? So when we've seen this data, all the senior guys here, we all came to this field because we said, if we are not going to change aging, you're not going to have any effect on all the age-related diseases. That's the only way that you can impact those lines here. I'll tell you, but since then, I'll let you in a second. Since then, we believe that we are right, and this is because of the great success with cardiovascular disease. Because cardiovascular disease, we can prevent better by medication, lifestyle, you know, aspirin, whatever. And people who get myocardial infarction, they get stent, they get bypass, they get drugs, so they survive. So you know what happened now that we know what happened to the people who survived heart attack? Within 2 years, if they don't get the second heart attack, they die from cancer, diabetes, Alzheimer. Because we never treated the aging part. We treated specifically just one disease. So now, if you're the guy who's going to age biologically faster than chronologically, and you got a heart disease, you're also most likely to have the same risk factor to get the Alzheimer or the cancer or something else. And this is what really worries me even personally, because I'm doing everything I can to prevent heart disease, and in my mind, I'm going straight to Alzheimer's. And I don't like that. The second point that is very interested in aging is, and it's a cartoon that depicting a point I want to make, but on a scale of youthfulness, okay, we are many investigators in the field of aging. Half of the investigators in this field in the United States are in this center, pretty much. And all of us have our favorite thing that changed with aging. But by the way, when I say youthfulness is decreased, you can measure things that are increased and things that are decreased, okay? It's just the directional of youthfulness. And we all know, and that's where I'm going to distract you a little bit, we all know about sex hormones that decrease with aging. We all know about growth hormone that decrease with aging. And then each one of us has one of those other lines, okay? Why am I showing you? First of all, when we look at this decrease of youthfulness, a lot of what we see here, hundreds of lines, can be really a cause or one of the causes of aging. And it's going to be important. The second possibility is that you can see all those with aging, but it doesn't mean that in our lifespan it's going to get us to any problem. You know, lipofuscin is something that happens in aging cells, senescent cell, things like that. I don't know that people die because of lipofuscin. Okay, it might be a good marker for aging, might be everything, but maybe it's not the cause of aging. But this is not what we are worried about. What we are worried about most is the fact that some of those lines here are protective mechanism of aging. In other words, if we start to get the breakdown and the changes in aging, just like when we have infection, we have inflammatory response. When we have aging, we're going to try and protect against those changes. And how do you know which of the things you're measuring is a cause of aging versus their adaptive or stressor or modifiers of aging? Because if you think it's a cause of aging, then to change it back to normal will be terrific. But if you think it's protective against aging, then changing them, or you're going to get into lots of trouble. So let's see, for example, what happens in, in elderly people. One of the things that happen, and I'm just showing a slide to summarize like a hot thing that's going in the aging field, and that's the field of senescent. And what I'm showing you is a pattern of secretory peptides in yellow in senescent cells versus pre-senescent cell. And the yellow means they're overexpressed or over-secreted. And it's from Judy Campisi. And the only point I want to make is that those senescent cells that secretes a lot of cytokines and other things are changing the environment of the body. So the body of young people and old people it's not the same platform anymore. There's a lot of biological differences with the example of those peptides. And I chose these and those secretory peptides because there's a science paper recently where if you take out the senescent cell in a certain model, then they become kind of youthful again, because you kind of change a lot of the environment. So why am I telling you that? Because there was a thought several years ago that the best thing to prevent aging in women is to give them estrogen. There was a lot of literature associated the estrogen with aging. Now, when you look at those things, you say, and maybe that estrogen will pretty much delay the aging process. Well, there are three problems with this approach that we have just to remember to whatever we are doing. The first thing, it's a very unidimensional approach, which we in the biology don't accept so easily anymore. Why didn't we also replace growth hormone or IGF? It also went down, and all those 100 other things. We just take one thing and we want to change it. By the way, even if you believe in estrogen, you believe in estrogen because of women, okay? But men age too, right? So you don't really think that estrogen is a major aging thing if women and men are aging, and they're aging in a lot of similar ways. So, but one problem is the unidimensionality of the experiment. Second thing, I already told you that there might be new interaction. You give estrogen on 70-year-old. Estrogen is a youthful hormone on a 20-year-old. But the question is it youthful on a 70-year-old body? When there are new interaction, they have more senescent, they have more cytokines, they have things. And then the third thing, we're also assuming that if there is a lower estrogen, it's not because it actually has a protective role. But is it possible that in all the physiological milieu, actually the low estrogen is protective rather than it's the cause? And I'm raising all those possibilities because estrogen was given in this study that was called the Women Health Initiative. And when estrogen was delivered, there were certainly some great effects, for example, on the skin of those women. But the study was stopped because estrogen replacement to postmenopausal women increased their incidence of cardiovascular disease, there was cognitive decline, and there's breast cancer. And in fact, since estrogen is not given to postmenopausal women, the incidence of breast cancer in the United States decreased by 25%. Okay? And so, if anything, estrogen is a pro-aging hormone and not an anti-aging hormone. And I'm telling you that, and I put those slides because I heard today lots of other examples. And I think Jim has really highlighted the fact that insulin resistance occurs with aging, but it doesn't mean that it's only, and it's not good in humans. We have very bad beta cells. We don't react very well. You know, it's not so good. But it doesn't mean that insulin resistance is not a protective mechanism. And it's possible that if you take insulin resistance away in certain organs, it's not going to be good because insulin resistance is a protective mechanism. Testosterone, just like estrogen, now retrospectively it doesn't make sense, you just replace estrogen. There's a trial now to give testosterone to elderly men, and I don't know that people have thought through it very, very good. And then, you know, there's other things, the oxidative stress that we always thought is a cause of aging. We find hard to prove it. And in fact, there are some models where the more oxidative stress you have and you can demonstrate, they live even longer. So things are really complicated in the biology of aging, but what we shouldn't do, and we shouldn't let the clinician do, is pick one thing and say, let's correct it, and that's the end of everything. It's not going to happen. Okay, so now I'm getting more to new science. And I think what I'll show you as part of the talk is that until quite recently, and many of the clinicians will still say that's it, that all the glucose metabolism is controlled by those 4 organs. In other words, if glucose goes high, insulin will be secreted and suppress glucose production by the liver. It will increase glucose uptake by the muscle and will decrease lipolysis. And metabolism will be normalized after meal or after glucose tolerance. And what became, and that's what it does. And what became clear in the last years is that the brain also has an important regulatory role. Both insulin and leptin are regulating glucose metabolism. IGF-1, I'll show you in a second, regulate glucose metabolism. And so the brain is exceptionally important to consider overall metabolism. And with that, I'm going to take, before I go back to showing you what's in the brain, I'm going to take a break here and take you through an extremely, I think, exciting thing that's going in my lab. And I offer people who are interested in something new to call me, because if you'll have a wow factor when I'll show you that, there's a lot to do. So what I'm going to talk with you are about mitokines. And really, this is a misspelling of mitokines. It should have been with a K, talking about kines that are secreted from one place and going to another place. And the reason I kept the CH is just to remind you that it's from the mitochondria. It's really mitochines. But the data that I'm going to show you are from Einstein and UCLA. And I have to disclose here that I'm consultant to this company, Cobar, that is producing those mitokines for use against aging and age-related diseases. So this is the hypothesis that we were following. So the mitochondria makes 13 peptides that are important for mitochondria. Okay, that's all we knew that they are producing. And there are numerous nuclear peptides that are delivered to the mitochondria. So the mitochondria has something like 1,000 peptides, but only 13 are from the mitochondria, and many others are from the cell. And there's a lot of apoptotic signaling and other biological outputs that are coming from the nucleus to the mitochondria. And so the question is, isn't the mitochondria talking back? I mean, what other examples you know of basically just one-directional effect? I mean, if the nucleus is talking like that to the mitochondria, what is the mitochondria saying to the nucleus back? What is it saying about some signals to regulate, for example, those proteins? What does the mitochondria do about going and talking with other cells of the body? Because if they're so important in energetic, they should be able to somehow communicate metabolic and survival signals to the cells. And even more than that, and that's where, when I'll come back to the brain, they should be able also to communicate with the brain where it's the central for energetic and metabolism. So the hypothesis is, what have we been missing? Why aren't we talking about how mitochondria communicate back? And several years ago, we've looked just for open reading frames across the mitochondria, and we found in a very simple screen that there could be probably close to 80 peptides that could be transcribed, translated, and that they would come from the mitochondria. And we thought, well, if that's true, and if there are peptides that are coming from the mitochondria, and we hypothesize that they have to communicate to the cell, to their own cell, to the body, then what does it take them to be mitokines? Well, they should be produced. They should be produced in the mitochondria. They should have biological action that is significant to something that we are interested. They should be secreted because we expect them to communicate with other cells. And so the communication with other cells. And of course, from an aging perspective, we think that they should decline with aging because so much of what we know about aging is linked somehow to mitochondrial function and probably number or biogenesis. So the first mitokines that was discovered, and was discovered by 3 labs at the same time, was named Humanin, and you know that you see that it goes back to other species. And one of the important first clones came from areas of Alzheimer, of human brains, where they looked at what's different between areas that survived Alzheimer and areas that were affected by Alzheimer. And they cloned this Humanin. There are several nuclear homologs that exist, but actually all of the Humanin is a mitochondrial form. And that's really where the source of this peptide is. It's a secreted peptide, and I'll show you that you can measure it in high concentration, both in CSF and plasma. It has high tissue expression in brain and testes, but it's expressed everywhere where there's mitochondria, which means every cell. It interacts with IGF-BP3, and there's a technical issue about it that I'll talk with you soon. And if we depict the mitochondria, the whole genome of the mitochondria here, it really comes from the 16S ribosome of the mitochondria, where there is an open reading frame. And as you see here, we are working on 6 other peptides, which we named Schleps, Hassi Cohen and myself, for small humanin-like peptides, or just because it sounded like cool. And I'll show you at the end that each one of those Schleps is actually transcribed, translated, secreted, and has some interesting biological function. So it's gene within a gene. It's transcribed and translated actually it's transcribed in the mitochondria, and at least humanin is translated in the cytoplasm. Okay, what do we know about mechanism of action of humanin? Let me say that it's a little bit complex. I'm showing you 3 possibilities. They can all exist together, and maybe there are some other things. But there's strong evidence that there's basically a cytokine, a trimeric cytokine receptor that has part of the ciliary neutrophilic CNTRF receptor, the WSX, which is the IL-27 receptor, and the GP130, which is the IL-6 receptor. It's a very unique trimeric receptor for the humanin, and it activates, like a lot of the cytokines receptor, STAT3, and it has some metabolic and neuroprotective effects. Also, another receptor was cloned. It's the FPRL1 and 2 receptors, and it looks to act more on the ERK pathway and be involved in calcium mobilization, anti-inflammation. And I think part of the effects of humanin are depending on where those receptors are expressed and which receptor is expressed more than others, which kind of complicates a little bit understanding the biology, but not actually the physiology of humanin. And then humanin itself is a partner to BAX and has an anti-apoptotic effect, and this is intracellularly. So the humanin that is produced here can immediately send an anti-apoptotic signal in the cell, although we have some evidence to say that there's interaction from here also on the anti-apoptotic activity, maybe not through BAX. So I'm going now to tell you a little bit about what we know about humanin and show you some of our own data here in blue. So humanin inhibits short-term memory disorders by cholinergic antagonistic scopolamine in mice, prolongs by 10% the survival of ALS model, prevents amyloid beta-induced memory loss in mice, shows to protect against experimental stroke. There are actually more data now on Alzheimer prevention in a double transgenic, triple transgenic model. But I'll show you how humanin normalizes blood glucose in Zucker diabetic rats, how it prevents diabetes in NOD mice, how it delays arteriosclerosis in ApoE mice, and reduce infarct size. And so the first data is just simple, and it just depicts one experiment that was published and was very easy, just taking Zucker diabetic rats with glucose of 300, giving 1 dose of humanin, and looking how glucose is normalized. So basically, it's an anti-diabetic effect. I'll show you a little bit more physiology when we get to the brain. The second thing that it does, or one of the things by which it does the glucose-lowering effect, is it increases insulin secretion. And I won't explain the protocol, but this is hyperglycemic clamp, where we get the glucose up to the same level in all rats, and to some of them we give humanin, to some of them we don't give humanin. To those we give humanin, their insulin level is much higher. So for a glucose stimulation, for the same glucose stimulation, insulin secretion is increasing, and we can get the same effect by using beta TC cell, C3 cells that are beta cells, and showing that humanin increase insulin secretion, and we followed it in many other models. Yes?
Okay, so that sequence, I see that it has a 1 cysteine residue in your human sequence. So that cysteine, if you replace to the serine, so does it reduce the effect of this?
We are doing that now. It's a good question. So how do you know to ask those questions?
Because I was looking at the sequence, and then I found.
Are you a peptide biologist?
I'm biochemist.
Yeah. Okay. So it's a good question. We're doing that. I'll give you an example of what we're doing, and it'll make it maybe clearer. This is an experiment where in Hassi Cohen's lab, they took NOD mice who usually develop diabetes by 20-25 weeks, and with the injection of Humanin once a day, decrease the incidence of diabetes, and also improve glucose tolerance test in those mice. Seems like an anti-diabetic effect. With our Nir Lerman in the Mayo Clinic, yeah.
If you have that NOD skin mice or mice that don't have adaptive immunity, does the Humanin still work?
Yeah, we think that the major role of Humanin here, I mean, there are two roles. One is it's anti-apoptotic. Okay, so it doesn't deal with the immunity so much as doesn't let the beta cell commit suicide. The second effect is glucose tolerance, because it improves glucose tolerance by insulin action. I'll show it later. And so glucose tolerance is improved also. So Amir Lerman gave a Humanin analog for 16 weeks and looked at the development of arteriosclerosis as a scrambled peptide and at Humanin. And it prevented arteriosclerosis in a major way and also improved endothelial function. So another kind of very unexpected role of Humanin. And lastly of the data I'm showing here is, when we've done it at Einstein, is we took this model of reperfusion after coronary ligation, and whether we gave Humanin before inducing or after inducing the ischemia, the infarct size have decreased in a really very significant way. So it's also reducing infarct size. So I went fast through, I gave you one slide for many data points to really show you that Humanin is one of those peptides that has lots of effects, lots of good effects on several systems we were trying to look at. And interestingly, in Hassi's lab in UCLA, they developed an ELISA for Humanin, and Humanin seemed to have decreased, to be decreased in Alzheimer, in CSF of Alzheimer patient. It is higher at Amir Lerman's in the Mayo Clinic in people who have normal endothelial function versus those that have poor endothelial function. As far as aging, Humanin decreases by 30% by age when we between 50 and 100 years old, but there is a really big drop between young and older people. So young people have really high levels of Humanin, decreases after puberty and further decreases with aging. It's a little bit similar maybe to the IGF story in humans. So I'm going to digress for a second from the Humanin, because I'm going back to that because I have to tell you one of the most interesting story in my mind, and that's how it communicates with the hypothalamus. But let me go back before I do that and show you some interesting thing. I already showed you this slide of how we thought that glucose is regulated by this, but the brain is important. And I think from an aging perspective, there are several things that we have to understand. The hypothalamus is resistant to insulin and leptin effects. So although insulin causes a decrease in appetite and does a lot of hepatic regulation of glucose production and other things, it's not as effective in elderly. And we published several papers showing that leptin, that aging is a leptin-resistant state. Even caloric-restricted animals do not react to leptin when we give it peripherally or centrally. So in a certain way, we have to think of other ways than insulin and leptin to repair the metabolic deterioration with aging. We have published a recent paper where we showed that when we give resveratrol to the brain, it regulates peripheral glucose metabolism. And we can stop that by actually giving a SIRT1 inhibitor in the brain. And then we don't get any hepatic effect. And we can also stop it by giving resveratrol to the brain by doing a selective hepatic vagotomy, and then resveratrol wouldn't act. So we believe that a lot of resveratrol actions on glucose metabolism resides in the brain. Not only, but a lot of it resides in the brain. So I just mentioned them passing, more to tell you why we're not looking more at insulin and leptin apropos aging, and the fact that resveratrol, we already done the studies we wanted to do. But I want to talk about IGF, which has been an interest here for a while. And I want to show you experiment, and I'll show you what we're doing with those experiments. We're using rats, and we are introducing cannula to the rat. We can do it either ICV, which we usually do initially, or we can target any specific nucleus in the hypothalamus, where there are very important circulatory network. So we do this surgery, young, old animal, 21 days before we want to study them. We let them recover from this surgery where they have a catheter out here. I have no idea that it's happening. And we put an IV catheters both in the jugular and carotid artery so that we can have an access 7 days before the study. And during the study, what I'll show you, we either infuse CSF or IGF with CSF, and we're doing lots of metabolic and insulin clamps, somatostatin, we give glucose, and I'll show you what we're doing. So, it looks like that. The rat is chronically catheterized and unstressed. And I know that you guys are doing here, you're doing mice, right? Or rats too? Rats too. So it's easier to do rats, right? You need less, and they're less, so they're chronically catheterized, they're unstressed, and it's only when you start taking pictures that they start complaining and they want things to do and stuff like that. But it's a really good study in rats. I'm looking forward hearing how it's going with mice, but rats are much better for physiology. And why IGF? Well, because IGF, if anything, IGF receptors are increasing in old brain. So the IGF is unique in the sense that leptin goes up because there's resistance, insulin goes up because there's resistance. IGF goes down not because there's resistance to IGF, it goes down because it just goes down. So there's enough IGF receptor in the old brain. In fact, they might be a little bit upregulated just because there's so little IGF in those old rats. Yes?
The IGF-1 receptors are which types of cells they are on? Like, could they be on astrocytes that are proliferating? Yeah,
that's an excellent question. Yes, we know a lot about that. If we have time, let me answer more. We are interested more in the IGF receptor that are in the POMC or the AGRP neurons. And we're doing lots of studies like that on that I don't want to confuse anything now, but I'll be ready to go, and I have in other forms some of those pictures. But you're absolutely right. That tells you there are IGF receptor in the hypothalamus. It doesn't tell you where are they, what's their distribution, and where is it important to have those receptors. But there is one thing that we know, and that's the important part, is that when we give IGF and look at FOS activation, okay, so we see if there's actually really cells that are being light up with IGF, then we have activation of IGF. And if we give a specific inhibitor for IGF-1, that is the JB1, we don't get, or we get much less of this activation. So the point is that there is IGF activation. The complexity here, and the reason we saw JB1 is because if you give enough IGF, you can also activate the insulin receptor. Okay, there's the IGF receptor, insulin receptor, and hybrid receptor, and they all have different affinity to IGF and insulin. We did a lot of studies to try and find the right dose that we give that will be IGF specific and not activate insulin receptor by just trying to see where JB1 blocks the FOS activation.
Which species IGF are you giving to the rats?
Sorry?
Which species IGF-1 are you giving to the rats?
Oh, we give human IGF. We give the human IGF mainly because we want to monitor also if IGF goes to the circulation and things like that. Correct. You know, I'm paying for not being deep enough now, but I want to tell more stories, but that was a good question. Okay, so now we give the IGF, and what I'll show you, because it was published, this part was published, is when we looked at glucose infusion rate, with and without IGF, remember it's all small IGF levels in the brain, we get activation of peripheral glucose metabolism, okay? And we've published that. Nick, in order to prevent a question, the story of whether it's hepatic or central, hepatic or peripheral is an important issue here, but I can talk about it later. But the point is there's a lot of activation of glucose metabolism by giving IGF to the periphery, okay? So remember, it's insulin clamp. There is an insulin peripherally, and what you're changing is IGF concentration around the brain. You get 44% increase in glucose utilization. When you do the same thing to all the animals, you get even a greater increase in glucose metabolism. So really what I'm trying to show you is, you know, there's lack of IGF with aging. You replace IGF in the brain, or I don't want to say replace, but you give a little IGF to the brain. There are receptors there, and you get peripheral effect. So we never looked at it like that IGF has peripheral effect on glucose metabolism, but it has. But, you know, that's really not the most important part, because the brain is not only the regulator of glucose homeostasis, it's also the regulator of energy homeostasis. And so our question for IGF was, what does IGF do to energy expenditure and to body fat distribution? And of course, then what we need to do is chronic studies, okay? Because we want to see things that are measured not in hours like in the clamp, but longer. So the chronic studies are in young and old animal, where we did pretty much the same protocol, except that those were 14 days protocols, okay? So it's protocols where we infuse human IGF, ICV for 14 days, okay? And the first thing we look at is at food intake and body weight. Looks like nothing happened, okay? Then we looked at what happened to body fat and its distribution. And what happened is that this infusion of central IGF has resulted in a large decrease in all the adipose fat tissues. In fact, there is also a non-significant increase in several of the muscles that we weight. But in order to get significance, we just have to do too many experiments. But if you're asking, you know, they're the same weight, yes, there's less fat, but there's a little bit more muscle, we believe. There are several other things that happened during the experiment. First of all, leptin levels in the young and old animals have decreased, reflecting probably the fact that there is less adipose tissue. IGF level has decreased in the periphery because when you give IGF to the brain, it regulates growth hormone, and then peripheral IGF goes down. So we actually had an exchange there. Okay. So I gave you those data, which are, by the way, are a large part of what my lab is doing, and we're doing it for a specific reason that I'll get later. But before I cut it again, I want to go now and talk about those mitokines, because the hypothesis is that those mitokines have to talk also with the brain. And so, again, the reasons are that the hypothalamus is where everything that seems important for mitochondrial metabolism could reside. And basically, first I want to show you that with aging, there's a decrease in humanin expression in the hypothalamus. That's in rats, and that's not in the hypothalamus, but in mouse brain. So the point again, it's very much of an aging mechanism. Now, before I show you the results of the humanin, I want to make one point, and this was published several years ago, but you saw that IGF, when we infuse IGF to the periphery, to the centrally, it improves peripheral glucose metabolism. It actually increased glucose uptake, decreased glucose production. But IGF in the plasma is bound to IGF-BP3, one of the carriers protein. IGF-BP3 is known to be associated with insulin resistance. And in fact, when we give IGF-BP3 periphery, it induces insulin resistance. But here we give BP3 centrally, and it induces insulin resistance at the hepatic level. In other words, the ability of insulin to suppress glucose production is pretty much lost when we give IGF-BP3 to the central. So I just want to recap this point. IGF centrally is improving peripheral glucose metabolism. It does it centrally or peripherally. BP3 does the opposite. So IGF that's bound to IGF-BP3, it's like you know what, you don't know how to deal with it really, and BP3 causes more of a resistance. And why am I telling you that? I'm telling you because when Hassi Cohen in his lab in UCLA cloned the humanin, it was a partner for IGF-BP3, and Hassi came from the binding protein fields initially. So it was binding to, so IGF-BP3, humanin was bound to IGF-BP3, and he went and found that the amino acid, which was important for this binding of the humanin and IGF-BP3. And he changed this peptide, and now this peptide is called, humanin is HN, and the peptide that was changed was F6A. So this switch of F in the 6th position to A allowed, you see here, humanin is bound to, BP3 is bound to IGF, BP3. IGF-BP3 is bound to IGF. IGF-BP3 is bound to humanin, but it's not bound to F6A, okay? And so we have a humanin that binds IGF-BP3 and a humanin that doesn't bind IGF-BP3. And it's important because when we give humanin peripherally, then humanin improves insulin sensitivity in the liver. HNGF6A improves insulin sensitivity on the liver even more, maybe because BP3 has a modulating effect. But I'm showing you here what happens when we do the same experiment. Instead of peripheral, we give it centrally. And remember, because we give it centrally, we don't know the doses. They're not matching. But you see that humanin significantly decreased glucose production, and HNGF6A is like this thing. We haven't ever had experiment in rodents where hepatic glucose production was close to zero, but it's a very strong suppressor of glucose production. I can also just share with you that that a lot of the activation that's going on in the hypothalamus are through STAT phosphorylation, also AKT phosphorylation, but not AMP kinase. And that because it's STAT, because we think STAT is the major thing, what we did is we repeated the same experiment. Remember, it's all ICV. So without humanin, without humanin, or with humanin, hepatic glucose production is suppressed, insulin action is improved. But if we do the humanin with a STAT inhibitor, we are negating the effect of humanin, suggesting that STAT3 activation in the hypothalamus is important to initiate whatever we are seeing. The SHLEP6 is different in the sense that it's a very strong tumor growth antagonist. And I'll show you one slide about it. Before that, I'll just show you an example. For instance, looking at apoptosis on beta cells with humanin and all the SHLEPs. And you see some are mildly protective, some are very protective, and some are, the SHLEP6 is not protective is pro-apoptotic to beta cells. Okay, so, you know, it's like the way that we do those studies. We take all of them and see what's happening, and they are very different in their biological activity. So humanin levels is increased in the growth hormone knockout mice. And in fact, in growth hormone transgenic mice, it's decreased. So if you wonder if there's more connection between humanin and the growth hormone IGF axis, there's more connection, although I cannot do a story out of that. I really use the IGF story to tell you about central mechanism and to lead you to the humanin effect on the brain. But let me tell you a little bit about the IGF, and you've done studies here on the growth hormone, certainly on the IGF receptor recently. You haven't produced, when I say you, I don't know if anybody is here, but you guys, you in the center. The IGF receptor that you tried versus Holzenberg, it wasn't as prominent in inducing longevity. But the growth hormone IGF axis has been important. And in our centenarians, we found that 2% of our centenarians, actually Yushin So, who was here, a human geneticist here, found those 2 mutations in the IGF receptor that are functional deficient, and people with this mutation have higher IGF and they're a little bit shorter. It's not the reason for exceptional longevity in humans. It's not the major reason. But in those 2%, maybe that's all it takes. Maybe they were protected from tumors. But the problem when you translate from animals to humans is the following. We know that IGF level, high IGF levels, are risk for all those cancers in many studies. We stopped here, I think, at 2002. But all the studies are pretty much the same. There is a risk between IGF and cancer. And this is maybe consistent with the animal models because a lot of the animals are dying from cancer. But what happened is that high IGF level, the same levels that are bad for cancer, are protective against osteoporosis, impaired glucose tolerance tests and diabetes, and ischemic heart disease. And I think that's the biggest paradox and the biggest problem when we come from the animal and try to talk with the clinician and see where we're going next. Because, you know, we'll take a bunch of people and they'll say, oh yeah, IGF is really bad for cancer. There's an IGF receptor antibodies that produced against cancer and they're in clinical trial. And the diabetologist and the cardiologist will tell you IGF is really great. So how are you going to work this thing? So our view is that we need to optimize the effect of IGF. It's not so simple to say deficient or not. We have to try and figure out what's going. And in our mind, the central effects, okay, if we could get IGF higher in the brain, we could improve insulin sensitivity, we could improve body composition, lots of studies of IGF improving or related to cognitive function. On the other hand, if we do it in the periphery, we get cancer. So we basically are thinking, how do we shift IGF to the center and kind of inhibit it in the periphery so that we can really get the full advantage of longevity that are not so relevant in our rodent model. And what we've done is we created, first of all, a humanized rat IGF receptor model. Okay, so humanized rat, because as you've sensed, mice, to do a good metabolic studies with mice, you need a lot of them, and I'm not sure what we're doing. Rats are just a better platform. Also, it's new because there's not much data on rats, certainly not on IGF, but even growth hormone and stuff. There's not many rat models. And the models that we have is humanized IGF receptor. The IGF receptor that I just showed you of centenarians, you know, the longevity IGF receptor. And then we disrupt one of the alleles of the IGF receptor to get an heterozygous. Okay? Okay, so this is the platform, and maybe this is a new model on its own, but that's not why we're doing that. We're doing that so that we can induce IGF in the hypothalamus, but by infusing lentivirus into the hypothalamus in order to get greater IGF effects on the hypothalamus. Okay? We are, and I'll talk about that in a little second. The second thing is we're using an antibody. By the way, Amgen, I think, are not in the game anymore, and probably we're using Merck antibody, but anti-human IGF receptor antibody that is in a clinical trial. And the idea with the antibody, or what we do with the antibody, you inject them only once. It's once a week injection because it lasts long. It blocks the IGF receptor in the periphery. It doesn't cross the blood-brain barrier, so that we're maintaining IGF in the brain, but not in the periphery. In fact, it's going to be, we predict it's going to be higher IGF in the brain because you block the IGF receptor in the periphery, there'll be a feedback loop to the brain. Okay, so that's the periphery. And here we'll have a permanent or temporal control. Here we block only the periphery, weekly dosing. And what we're going to look is, of course, longevity and also all those things of healthy aging, some of the things that you're doing, some of the things that we're doing. So you know, we're thinking basically, we're creating here a new humanized model that's kind of relevant to what we know from human physiology. And we're going to shift, by several experiments, we're going to shift the IGF to where we don't want it to where we do want it, and hope that this will have a better effect on the rodent and that we can sell it then to the people who are doing human studies. So again, I want to just end here and start having a discussion about what we heard, and just have those names all who had major parts in what I was showing. So thanks for listening now, and ready to take questions.